Thanks.
Okay.
That's it. I just wanted
Come
to see
wide,
for a
please.
minute there. I thought he was somebody different. This is in that genus we call Poliopogon right now, and we collected one of these earlier to try to get a better handle on it. And now we'll take a look at this rock.
Okay, so it looks like you have a question from Steve.
Okay, let's see. Yeah, so Steve is asking, what about these planar Chrysogorgia colonies like Chryses? And I don't know. I mean, when you look at an area like this, you have such a mixture of planar corals and then these spiral corals, and then you have the small little anthomasses. I can't do it. Heteropolypus. So it looks like not one size fits all. I mean,
no, very different strategies.
Corals are doing pretty well regardless of their shape. You know, the planar corals, of course, they'll often orient in an optimal direction. They'll put their biggest face right perpendicular to the current. And you can see this a lot of time with the planar corals, that they actually are indicators of coral current direction.
Yeah.
We can see along this one, it's mainly across the slope, not up and down it.
Yeah, exactly. Perfect. You know, something like that. But I don't know. It's a really good question. There's just different solutions to successfully feeding and getting established in these places, I guess.
Oh,
yeah, look at this one, perfectly horizontal, so the currents come along the side of this rock probably. These bottle-shaped ones, they're very flexible too, but then, you know, they can move around. A lot of these Chrysogorgics
Look at how they're leaning into the current. It must get pretty strong up on the top.
Maybe we should get a zoom on this potentially bubblegum, because the polyps are out compared to that last one the polyps were in.
Hey, watch this, Andy. It's not a bubblegum, but we'll let him zoom in on it.
Bubblegum-esque. Is this not the same one we just sampled?
No, it's not the same one we sampled. Okay, go ahead. Zoom in. No problem.
Go ahead, zoom in.
Yeah.
Tell me that's different than what we just sampled.
That's different than what we just sampled.
So different, Trevor.
That's why I'm asking you to tell me that. Thank you.
I'm not even pretending no coral, and I could say it's totally different.
Yeah, lay into them there. That's good.
All right, you want me to keep stepping?
It was worth. You're going to win one of these, Trevor. I guarantee
you.
It.
Keep going, step. Yeah. If I just keep saying it enough, one day I'll be right.
I know.
Here's a stalk.
Here's a stalk. Yes.
What's at the end?
It's not a gigantic. This is the bolosoma again. And if you come up the back side of this, you'll see the big osculum on the top of it.
Osculum.
Osculum. It's a hole. The water comes into the sponge and then it exits through these holes.
Bridge nav, another step, same bearing, 30 meters, 151.
This orientation gives a look.
Okay, zoom in, please.
And where are you? Let's see. It'd be a little bit higher if you can pan up a little bit.
Coming up.
Well, it's still a bolosoma, but I'm feeling like a liar right now. Where is that?
Uh-oh.
Wow.
So the characteristic of the bolosoma is that a hole at the top, you
Yeah,
said?
you're seeing a little bit of a depression on top, but usually they have a very discrete hole right above the rim. So telestrator, since everybody can see this now, but normally they'd have one right there.
Oh, yeah. That's the exit wound?
That's one place where water can get out. Huh, that's pretty weird.
Okay, come wide, please.
I'm going to let it go, but it's still bolosoma. Convinced it just might be a little bit different one.
It's a nice rock.
It's a nice rock, yeah.
What determines the colors of the corals at this depth, Chris?
I can't hear you. I can't. Boy, I don't know. Only
chemical structure.
That's what I'm assuming, that whatever types of compounds they have in their tissue, they reflect this artificial light differently. Some will
and whether
absorb
it serves
a lot
purpose is unknown.
Yeah, it's unknown. I really don't know. With no light down here, you know, why do they have colors?
This one looks dead, mostly dead.
Yeah, it is. It's a bamboo, but you might find a sea star on it because it's usually sea stars that ravish these bamboos.
Yeah, very fat and happy sea star somewhere.
Yeah.
So can this recover if the sea star is left? Can it recover?
Yeah, it'll probably still keep growing out, and maybe some of the tissue will grow back down the stalk. But there's some not some algae, but there's a hydrozoans on some of those branches that might prevent the regrowth down the stalk.
Is hydrozoa the same as hydroid?
Yes.
Okay.
Well, hydroid is kind of a type of hydrozoan.
Steve's got a comment in there. He thinks we've crossed a diversity boundary. So we weren't observing a lot of these species down below.
That's a very good comment. And there is a more typical Anthomassus over to the left, not the 3 polyp ones that we were seeing earlier. This thing is there is kind of what we see more often with these red mushroom corals. That's got what, about 10 or so polyps on it.
Yeah, and it branches out from the almost like from the base.
See what's eating the stuff on the rocks. Yeah. Something's eating stuff on the rocks.
Should probably keep moving here so I don't fall too
We're
far
starting
behind.
to get some nice bamboos here, so the population is picking up, as
Yeah.
Steve was noting.
Size, too.
Yeah, see this, now we're getting more and more of these bamboos showing up. A good observation of Steve.
Is that just sand? It was
The
sand.
line, yeah, I think it's sand, yeah. Sediment, whatever you would call it.
So, well, small yellow colonies. I didn't see the small yellow colonies, Steve. I'll keep an eye out for them. He was just noting that in the chat line.
So we just passed them. Oh, there's another.
Oh, it
Another
could
note.
go down if you, the bottom right, excuse me.
Oh, I see it. Is that a crinoid?
Nope.
Ah,
It's anemone. Anemone?
Nope. Well, it could be. Yeah.
what's
excuse
that
me.
crinoid looking thing?
We haven't really seen many
Oh, you're looking at a
crinoids.
different thing. So you're looking at this guy,
Yeah.
which is a crinoid, a
Yeah.
feather star. I'm looking at that guy.
I thought you loved crinoids.
I do, but I like
Okay.
stocked ones, remember?
Ah, okay,
Well, I
fair
like
enough.
them all, but
Zoom in, please.
yeah, what I'm looking for is, are there tentacles right in that center of that? It's really dark, but can we
Okay,
see
come
a
wide a bit. I'm kind of nestled in. Come full wide and I'll get a little closer. And we can get a good answer to that question. Science pressing question. Okay, zoom in, please. You can go all the way.
I am
Ah.
not seeing an
Not see any. Nope.
inner ring, which means it's an anemone. It's not a tube anemone.
All right,
As a
sorry, I'm bouncing.
Cereanthids,
Come
the
wide.
tube anemones would have an
Thanks.
inner ring of tentacles right around the mouth.
All right, I should move along now.
Okay, thank you for that. That was good. So we do have these sort of sand-dwelling anemones, I think, in the guide, and that's what we call them, anemone, because we don't know what they are.
Oh, actinaria, potentially,
Have you on the picture?
like that.
Yeah, that one.
Bridge nav.
Yeah, that's kinda
30
one type of
meters,
them,
151.
You're looking
Color's
at the guide right now. Right.
Yeah.
Is that like a burying anemone?
What's that?
Is it a burying anemone?
No, it's
No.
not. It's just most anemones will attach to rocks, and then there's this weird
What's their
sort of
bearing?
brown one
150?
down there that kind of likes the
150.
sediment. Whatever
I'm not
reason.
stuck on it if they have any better ideas.
This is fine.
Yeah, again, these flat surfaces aren't the best for attachment.
Yeah, and there's a little more sediment dusting on
Yeah.
these too, so
What's that dark thing?
I don't know. Want to take a look?
Looks like a saxophone in miniature.
A what?
Never mind.
Probably
Whoa,
best.
that was out there. We're
I was
taking
just
a
going
look
by
at this thing,
shape.
and I think this is going to be a mollusk of some kind,
Ooh.
pluribranchid.
Okay, zoom in, please. It is a rock.
And
Oh,
yes,
it's a rock.
yeah, a pluribranchid rock.
Is it a
Uh-huh.
paragorgia?
What?
I think it's a paragorgia.
Saxophone rockaday.
Wow, that sounded pretty cool.
Boy, did we butcher! Boy, did we butcher that idea.
Oh
didn't
yeah,
we?
Wow.
we worked that one.
I'm glad we all got very excited for it, though.
But you know, that's kind of the game, to try to see if you can ID something from a distance, and then you zoom in and you see if you're right or not. That's also a way of learning.
Mm-hmm.
We were talking about that in the field, how many times we've seen log bears, where we're so sure it's a bear, but it's a log, and then it fools you multiple days in a row.
Well, that rock looked very different than all the others around it, so
Yeah,
it caught our attention.
yeah, it looked like it. There's a type of a dark brown mollusk that lives at these depths too, that it looked very similar to.
Here's a stocked something.
Oh,
Oh,
dang.
boy.
Oh, this is
sponge
Bolosoma species B. It'll have a yellow stalk on it, and it will have that osculum, or I'll eat my hat.
Okay, well,
Okay,
you don't
zoom
have
in,
a hat
please.
on,
We gotta
but
see this hat eating.
There it is. See it? See it?
Oh, yes,
It's there.
yes.
It's there. I'm going to circle it right
That
there.
hole.
See that big depression right there? That's what I was looking for with the other one.
Oh, yeah. Cool. Good call.
That's characteristic of this genus Bolosoma. That
So is the stalk hollow? It looks like it descends down into the stalk even.
yeah, I don't think so. I don't think so. Sampled these once before. They're really beautiful, and they're really classic with this yellow stalk. They're wonderful.
Wow.
Just all sorts of, that's a beautiful thing. Okay, come wide, please.
Oh, that's great. Bolosoma species B is what we call it.
Species B.
Thank goodness I don't have to eat my hat, because I only have one Nautilus hat.
Got some elevation.
Can you look a little to starboard?
What was the starboard?
Can you look a little that way? I'm just trying
Oh.
to
Yeah.
see if there's anything cliffier that we're missing out on.
Red thing down there. I don't know what that is below the lasers.
Oh,
that a
and
shrimp?
over there.
Oh, it could be.
Shrimp or one other one.
See anything that tickles your fancy?
That little red thing.
Little red thing.
It's
under the lasers.
Oh, yeah.
it's hard to see on the. Oh, there it is, right there.
Shrimp?
I think so, but we haven't really got any close-ups of too many of those.
I'd say that's a shrimp
That is a shrimp.
or a prawn.
No, it's a shrimp,
No,
and
it's
it's
a
a
shrimp.
long-legged shrimp, and I am having a senior moment here. I know what this is. It's open crying out loud. Let's see. Okay, here we go. I'm getting there. Yes,
Bridge nav, Other step,
nematocarcinus.
same bearing.
So that's the guy right there.
Oh yeah, look at the hump, the little hump on the tail.
Yeah, and the legs are really skinny,
Still
so you
150?
can't really see them. It has
Yeah.
really long, skinny legs, and
Not
there's a
loving it,
number of
though.
different species.
Just
So
don't
we'll
have any
just go
better
to
ideas.
No.
the genus right now.
It's definitely not following. It looks like we're staying on a contour, but I mean, based on the
Yeah.
upness.
We keep trying to go that way, and it's not. We can
Yeah.
try and go back up that way, or we can.
Is this
push
our
ahead.
data or
No, it's
Ooh,
not.
that's
But
why.
that doesn't matter.
That's
There's more. Is that more dead things on the right?
well, that's like that that spine
Faraday.
Oh
Faraday
yeah, the dead
sponge
sponge
skeleton.
skeletons.
Oh, okay.
one, Skeletor or whatever. Yeah.
They take a long time to decompose, these dead skins, and then once they got that manganese crust on them, I think they could persist for a really long time. It's like a coating.
It's like petrifying.
Petrifying
almost,
it.
kinda, right?
How long does it take to start the manganese crust where it's appreciable and noticeable?
Well, it probably starts pretty soon after the thing dies, but then how long does it take to even accumulate a film? I have no idea.
What's this?
Partially alive. One of these giant stalked.
It's not really
Arr.
alive.
No,
Is it?
it's not. It's looking pretty
Too bad.
deceased.
But that also
What's
means
to
by
the
that
left?
dead sponge that there's some of those particular sponges somewhere. Probably
Look at the upper left on the Argus view. Or is that a rock structure or is that a
More sandiness, I think.
yeah.
The white
sand?
stripe.
Sand?
Oh, yeah.
All right.
Andy, you're going to...
I know. I'm going for, what is it? Magnanimous? What is the one you've been trying to sell us?
You mean a Gigantea?
Giganticus, yes.
No, that wouldn't have been that. No. No, you're going down in flames, Andy. Little Chrysogorgia again. We see that seems to be a very dominant member of the community here.
It is.
Yeah.
So what is the reason why we don't see
Just keep
more
going south.
crinoids here?
I don't
I
know.
don't know. We're still going upslope, so I'm not too
But,
fussed.
there's still plenty of time. I'll bet we see some before we finish
That sound
this very
reasonable?
long
I don't know.
dive
What do you
that
think?
we're doing.
No, either.
What are we at
Turns
now?
out we don't.
20.
Yeah.
What's our depth right here?
Let's see. At 2,300. Little white thing there. I don't know if that's a dead colony of
coral.
stylastrid. I wonder if we could take a quick zoom on this guy right
He's coming up
there.
center right now? Yeah.
Yeah.
Go ahead, zoom.
Right where the lasers are.
Bamboo?
Yeah, it's dead.
I see the little nodes, though. Cool.
Yeah, you see how the branches are kind of pointy there? I think that's probably dissolution where it's actually dissolving.
Oh,
Okay, come wide.
That's a guess. I assume that's why they're pointed. There's another sponge. It was one of the ones we collected to the right, so you don't need to see that one again.
I don't even want to look at it. I can't remember the word.
oligotrophic.
Oligotrophic.
Nutrient poor.
How many points would that be if that word came up in Scrabble? I'm just curious. Probably not that many. I guess most of the letters are
Yeah.
there's a little Iridogorgia, and it's on its side, and so I think Steve was right about this one, that it's most likely Iridogorgia bella, which
There's a
typically has,
fish.
yeah, this one that's right there.
Did you want to look at it?
Sure, we'll take a quick zoom.
There's
Again.
a little
Pretty low.
fish right there.
Okay, go ahead and zoom.
We don't have to, yeah, so this is kind of the same view we had before, but in any case, I think he's right. I think this is probably Iridogorgia bella. It tends to have just a sort of a top knot of branches, and then the rest of the stalk is pretty
You can
bare
zoom
of
more.
branches, unlike some of the other Iridogorgia, and in very tight spirals.
Mm-hmm.
By the way, when the current's really raging, another
Okay, thanks, come on.
advantage of this design, it
Spreads
kind
out.
of stretches out.
Yeah, that makes sense.
Oh, darn it, I wasn't looking again. Okay, was it small or big or? Yell it from the hills, Aphrodite, because get a
Okay.
chance to see them. Couple of small black corals, probably that Alternatopathes over there on the right. We're just passing those guys.
Trevor, do you mind if I borrow bubble to check gauges?
Do it, yeah. Absolutely.
Quite a bit of marine snow deposit here. What are we doing on rocks? We good with rocks?
I guess the way I'm thinking of it is that we're kind of used to doing the 12-hour dives. This is the double. And so we've already got a bunch of boxes filled. There's no reason why we...
Oh.
And they've got some lower down, and I think they kind of want some a little further up to space them out. But these right here probably aren't going to be all that great for Meredith, but I was, again, thinking that... I don't mean Meredith, Beth. But I was thinking, I think she knows exactly what she's looking for. Should be plenty of opportunity for her to grab what she needs.
Mm-hmm.
Yeah, maybe we're hitting that curve now. Sure. I judged with how Argus looks too in the
Do you know the history behind the naming of this seamount, the hhmm seamount?
Do you know what it is?
No, I'm asking you if you know.
Not a clue. Yeah, I suspect it was probably a name of a ship, because a lot of these things are, you know, when you have Paul and Starling, and it's quite possibly it was the name of a ship that was, here in the Hawaiian Islands a century or two ago. It's another one of those supposed chrysopathes in the center, flanked by a couple of the gold corals, the chrysogorgids.
Oh, this is kind of a little rock mound we're on. Looks like something happened here. A lot of dead small corals and sponge stalks.
Yeah.
So are all these dead ones bamboo coral stalks, or are they all various types of coral?
I'm wondering, you know, I don't know. No, maybe it's kind of worth taking a look.
Okay, zoom in, please. Check this one out first.
It's going to be bamboo, I think.
Maybe. I don't see nodes, though. Do you see nodes?
No, but I still think it's a bamboo. There might... I still think it's a bamboo. It's just very weathered.
Okay, come wide, please. Look at a couple more.
Oh, it's rather large. Nice chrysogorgidae.
I guess I'm on a rock. Huh. Okay, zoom in there, please.
Yep.
Yeah, that's a bamboo. You can see the node, that little lump on that right-hand branch.
Okay, you can stay in.
Okay.
Another node, I think.
Yeah, I think so too. Probably right there, right where
you can stay in. I think I got one more just up here.
It doesn't necessarily mean there was a vent. These things could have died over a considerable length of time.
Okay, come wide, please.
Just one by one. You know, when we see a cluster like these, we immediately go to hurricane, earthquake.
Here's something cool we haven't seen in a while is a good-sized chunk of sand that actually shows the current ripples.
Oh, yeah. Interesting.
You really see it in Argus view.
Oh, I see it in the Argus view now.
Coming up in Herc view, yeah.
And what's
rather
the
large
orientation of that? Is that going east to west or something?
I'll line up to it. So this is going to be obviously 90 degrees out.
Right.
But, I'm looking approximately just shy of west.
So is it north-south?
Should be across the slope, which is what we were seeing.
Oh, across the slope, okay.
Yeah, the slope is pretty much ahead of me.
I have another question.
Okay.
Were these seamounts ever coral atolls? Did they break the surface at some point?
The one we're on right now, yes, it was. It's called hhmm. And it's what we call a guyote, which is a flat top seamount. Because, oh, can we zoom this? I'll
Yeah, you
I'll
bet. Go
finish
ahead
the
and
answer
zoom.
afterward.
Yeah.
But this is a different sponge that we haven't seen so far.
Oh, yeah, these are beautiful.
Look at the base to start, and I'll move up.
Sure.
Very translucent.
So this particular sponge is in the genus Walteria. It's in the family Euplectellidae.
Is that a squat
And-
lobster?
Yeah, a squat lobster in it, I'm pretty sure. Yep, that white thing, and it's also got some shrimp in it.
Shrimp, and then what's behind it? Is it a black coral behind it, or what am I seeing there?
You're seeing, I think they're hydroids that are growing on some of the lateralia, these long extensions of the sponge coming out.
Okay.
So we really don't know what this is. I think it's been collected, and I think we call this Walteria woolly because of all the frills on it and things. Does that look like that, or does that look like some other coral that's next to it? That
I can't tell.
I can't tell either.
I'll keep coming around. I got a little bit more. Hard to say.
So that white squat lobster is a Munidopsis. It's in the family Munidae. And I think we haven't seen very many of those either.
No, not on this cruise.
And then these shrimp, oh boy, let's see if, Steve, Munidopsis. Okay, Steve actually agrees. Steve, any idea about the shrimp that we're looking at? I don't think that's Bathypellinoma or something like that, but it could be. I think they got to have one bigger claw or something, if I remember right. Oh yeah, he said it could be Bathypellinoma or Bathypallimonella. So okay, we're on the same page.
Cool.
Great, thank you. That's
Okay,
a
come
new one.
wide, please. You don't want to sample that or anything?
No, we've got this one. It's a fairly common sponge. Thank you.
Bridge nav,
So-
30220.
So these flat top seamounts, they were a volcano that erupted to such an extent that they came all the way up from the abyssal seafloor, broke the surface of the ocean, and became an island. Oh, excuse me, we got to look at another one.
Oh, there's your stock crinoid?
No, it's not.
No, it's not.
No, it's not. Sorry.
You want to look at it still?
Yes, I absolutely do.
Okay.
This is something different. We don't need to collect it, but I know what it is.
Okay, zoom in, please.
Oh, yeah.
This is a sea pen.
I like that. That's pretty.
And this is in a genus called Umbellula, and they're very cool, as you can see. And it's got a big stump that's buried in the sediment, and that's how it anchors itself.
Cool, okay.
So they're really, really beautiful, delicate type of sea pens. In fact, that is a gorgeous image of one of these guys. They're called sea pens. You can see it there. If you look in the tissue down the stalk, it's got what's called a pen, which is a harder structure that runs the whole length of the stalk.
Oh,
And that's
yeah.
why they're called this type of thing.
All right.
And then we got another one of those nematocarcinus shrimp up in the sediment. I wonder if you could zoom that shrimp, because I think we'll be able to see the
Zoom
legs
in.
on that one with this angle. Yeah, really fine, thin legs.
That's my whole
You
zoom.
can see them sort of
Okay.
stretching out above it.
Mm-hmm. They're quite long.
Yeah, right in there, you can see their legs right there.
Wow. Yeah, very thin.
Yeah, that's beautiful. Yeah, thank you.
Okay, come wide. All right, I got to catch up now.
Okay. So this volcano was an island. It stopped erupting. Erosion started occurring. Reefs started growing around it. And these processes combined resulted in it topping, flattening off the top until it became a lagoon as it was sinking back to the ocean. And as it kept sinking, the coral finally stopped growing because it was sinking fast enough. And once it got below 200 meters, it was technically called a guyote. And then it just kept sinking. And that's what we're diving on. It doesn't look like a flat top because we're nowhere near the flat top right now. We're on the side of the seamount. But that's a very good question.
Yeah, thank you for the answer.
Another sea cuke.
Sea cuke.
Mhm. What is,
Nice looking rock, potentially.
Yeah, you know, those look kind of good, don't they?
Uh-huh.
If we can find a really nice angular one.
Angular one, and we'll sample it.
How about the one over to the right there? This one that's way on the right there. Is that a
Position.
possibility?
Bridge nav, hold position.
Yeah.
There's another hemicorallium, the precious coral, on the boulder.
Quite a few things on that boulder.
That too big, or will that work?
That's pretty big, but we got a big rock box open still, don't we?
Well-
Echo or Foxtrot still open?
yeah, we got E open.
Should we do it?
I say yeah.
All right, then let's do it. I think Andrea would agree with you.
Is that the biggest rock?
That's I'll tell you, I'm not carrying that into the lab. I'll give you a clue.
Andrea will.
She wants it. She's
She
got to
will
carry
run
it.
to it. Yeah.
Get one of those baby carriage straps, right?
Uh-huh. Like a baby rock Bjorn. Those things, baby Bjorn.
So the woolly sponge that we just observed, is that the first one that we have seen
Yes.
this expedition?
Yeah, and I'll bring up a picture for you so you can.
Jason actually pointed that out. Jason
What's that?
in the audience.
Did he?
Look at
Well,
that.
way to go, Jason. All
Yeah.
right. Where are my sponges here
What
in
is
this
not?
gallery?
Oh, 5. Yeah, okay. That should be good. I'll give you that on here. Yep, it's all ready. So this is halt, this is grip lock, and don't worry about the right one. So you're halted now.
So, well, this is kind of a partial view of them. That's sort of woolly. That's the tip of them.
You gotta press the top button.
And then that's it.
What's all of this?
Yeah, you gotta.
Yeah, those
you
are
gotta
little
want it.
stars
You really
that
want
live
it.
in it. I'm not willing to go to Lucardi on this thing. I'm just going to call it woolly, which is what we used to call it, because it wasn't clear to me it had some of these
No, you do not wanna
characteristic
press and hold.
oscula that that species has.
Oh,
No.
yeah,
And
you
what
still gotta turn
so
that
what
guy
is Bathypalm non
yeah.
That's a shrimp.
That'll do
So
it.
it's
Oh,
a little
that's the
red
shrimp?
yeah,
Now
that,
you can.
that live on
Yeah.
sponges, and particularly on sponges, I guess.
Oh, Chris, that's a pretty good sized rock you chose
Well, he said he could take it.
there.
And you can call for zooms if you need them or want them. You want bubble on the arm? Right.
A boulder, isn't it?
Mm-hmm.
Oh.
Almost boulder-like.
Okay. How about that?
And so again, we're looking for these angular rocks, or rocks that don't look nearly as weathered as the others in the area. Those are ideal for sampling, for aging of the rocks, to try to find out how old these seamounts are.
I don't think you can lift it.
No, Sir Kirk is.
Lock the jaw is the left button.
Oh, okay.
Do you want? Is that what you're asking me? Yeah, that's the left button. I don't think you got it. Yeah, I don't know if you can lock it when it's in halt. I think you can. Not that button. That's the wrist rotate. You want this left button here. Yeah.
There it
Oh,
is.
there we go.
Nice.
We have liftoff. Oh, we got a
Oh,
creature on
nice.
there too. If we could get an image of that creature,
Okay, your bubble over
possible
on starboard box. On 2 was the preset?
They're wanting an image as well.
Okay, there you go.
Trevor, they're wanting an image as well.
Oh yeah, so
I just want
right
to
in the
see
front
what's
there,
on it there.
yeah, right there. Hold it there, and I'll.
We got a
Okay, go
feather
ahead for a
star
zoom.
on it.
Mm-hmm.
I'm surprised he hasn't jumped off by now. Typically, they'll bail if they detect the threat.
Now, if you unhalt, you can give it a rotate. So use the rocker button by your thumb, yeah, and give it a nice slow rotate. Just push it very slowly, very gently.
What's that white dot
Push
on
it,
it?
push it less hard.
It's
Yeah,
sediment.
go slower. There you go.
What that white stuff is on it.
Nice. Lovely.
I'm going to go out on a limb
Right
and
there
say
on
that
the
this
top.
feather star is in the family Entodontidae. And mostly it's
What's
based
that?
on
That should be good.
just the shape, but also the feet, which are called cirri on it, are pretty short.
Yeah.
Okay,
There's
you can
a
stow it in the
sponge back
box.
there too.
I'll be ready to open it.
Spotted that sponge.
Oh, there's what's flying. Oh, never mind.
Starboard box.
I thought it was a fish, but it was just debris.
What do you want? Okay. Oh, wow, that was very helpful when that just went off there. Sorry. There you go. I'm actually going to do this.
Is that a big white sponge at the top of the
Ready for sample?
screen?
Yes, it is.
Here's sample. I'm ready on the box.
Yeah, ready. Sample is 033. Box E.
Okay, box coming out. Sorry, it was Echo or Foxtrot?
Echo.
Okay, that's
Sorry.
the close of the big ones.
That's going to fill that big one.
Yeah, absolutely.
He doesn't want to let go. He's like, I'm going up.
Ah, you're a little close. You want to be a little farther away.
The tricky part about this game is that you're looking at a 2D image, but you'd have to have awareness of the 3D.
One more time?
Yeah.
And it's kind of
Push
a
it
learned thing,
that
and
way.
these guys are amazing after
AKA
a while,
that
the way
way.
they can actually build in their heads a 3D space out of a 2D
Yeah, right there.
image.
That's good.
So
Yeah.
they know how
And
close
straight
or
down
far
from
they
there.
are.
And
Yeah, it's quite a skill.
I get it started in there first before you let go. Yeah, exactly. Now you can open jaws.
Bonk.
Well done.
Good
Ooh,
job.
yay.
All right.
Nicely done.
That was sample 033.
That was great, Antonella.
Can you repeat that?
done, Antonella.
033. Sorry.
033?
Yeah.
Okay, thank you.
Thank you.
All right. There's your regular
Well
salvo
done.
again.
Are we going to be able to get off the bottom now?
Yeah, we'll stow the arm and then be on our way.
I spy a sponge.
Is there a charge for oversized luggage like airplanes? Or
Yeah, definitely.
Good thing you guys are frequent flyers, frequent divers.
Well, you're in first class. You get your luggage free anyway. We're back here in economy.
Not very economical.
Right.
You can put it farther outboard.
On board, okay.
Yeah.
So we have another
good there, yeah.
question from the audience.
Sure.
Is there ever a concern of bringing
you
up
can
a
press the
venomous
blue
or
button.
poisonous animal?
Yes.
Okay, great.
And that is another great
Nice job.
question.
So
There are these big urchins that are called echinothurid urchins. And unfortunately, I did not know this, and the woman I was working with many years ago did not know this, and we collected one of these big, huge urchins. They have kind of small spines, but they have a disc that's about 6 inches across.
Yeah, I know.
And
Okay, can I have front
she
porch
just
on
thought
that?
it was a
We'll move
standard
along.
urchin like in shallow water, and she grabbed it with her hands, and she paid a price for it. Her hands swelled up, and they were sore for days.
Oh,
And
wow.
Wow.
very, very painful. And so we don't collect the kind of thirds anymore right now.
It's not.
That must have been scary too.
All right, I'm going to mess
It
with
was
Argus
scary.
for a bit. I
And
turned
she
Argus
hurt
away, so I'm going to catch up.
immediately.
You're
There
gonna
was
go
a
come off,
sponge
turn
right in
around.
our view somewhere. I don't know if they cared.
Yeah, it's right in the back, and it might have been the same thing we collected. It was hard to see. There he
Okay.
is right there in the upper right.
All right.
Mm-hmm.
I can take a quick look. I'd like to get going, though.
Sure, it's fine.
Looks like a morel.
Yeah, I think we can pass on this. It is the same thing that we collected.
Thanks.
So that was a very good question. The hydroids that you can sometimes see on sponges, they sting. I have personal experience with them by being stupid and handling some sponges we brought up with my bare hands, and I didn't think they were hydroids on it. And they didn't kill me or do anything, but they hurt. And not as bad as that urchin, though. But
How long does it last for?
probably a day or two.
Oh, that's unfortunate.
Uh-oh, I thought I saw Candidella gigantea, but that's okay.
Where? I can
Down
do it.
below you, to the left. You just passed over it. Keep going a little bit further, a little bit further. Right there. Right there.
All right. I'm still in fast mode, so it might be a bit bouncy, but go ahead and zoom.
Come on, be it, be it. It is, yes! Candidella gigantea, we found it.
So you can tell us the story now?
Yeah, you know why it's
sample it?
gigantea? No, we don't need it. It's a common one, thank you.
Okay.
You know why it's gigantea?
Come wide.
I have no idea.
Because the polyps are so large. The coral is so small, but the polyps are huge for this little stocky thing.
Okay.
Yeah,
Can you say the name again, please?
yeah, Candidella gigantea, and I'll bring that up. Ah, we found one.
Nice.
Let's see here.
All right, I'm pretty caught up now. We can get more ship moves in, or do you want to hang around for a while?
No, we can keep going.
Okay.
Now that we saw our Candidella.
Mm-hmm. We're done. Our work is
So
done.
this is a
Bridge nav.
coral or?
Could we call it Nelson?
30 meters, 220, please.
Oh, that's
Right.
a tiny Iridogorgia. Look at that.
Mm-hmm.
Got another one of these sponges. More of those Anthomastus or Heteropolypus. The red guys on the rock to the
Yeah.
left.
Mm-hmm.
So same characters we've been seeing through most of this dive.
Same sponge.
Yeah. What that frilly thing is over there. I guess it's Chrysogorgia, some kind of a planar Chrysogorgia.
That's a really big rock. Can I ditch a weight plate?
Oh,
I think so. I think that's in our permit, if I'm not mistaken.
Yeah, as long as we don't smash any.
Yeah, find
any
a clear
animals.
spot on the bottom,
Yeah, great.
which
That's
shouldn't be a problem. There's plenty of them.
plenty of clear spots. Yeah. Of course, right now we see a lot of life, so that's not going to happen. Rats, so much excitement. That was a big rock.
Just gotta, you know, get the more sponges to counter it with the buoyancy.
Yeah, exactly.
Trying. I'd like to see a little more variety than we've been seeing so far.
Yeah, it seems to be repeating.
Might as well get something cool to look at in the meantime. How about that big stalked thing?
Yeah, I don't know what that is. So it's.
As soon as I have enough leash to get over there, I'll park. We'll check it out. I'll dump a weight plate, and then we'll move on.
Okay. This is just the Chrysogorgia, just kind of a
look
big
at
one.
that big thing off to the left, on the horizon.
Yeah.
Oh yeah.
Oh, might be one of those sponges still attached. It looks like, no.
Whatever that is, that's big.
No, it's a coral.
I think it's one of those spiral corals, which I've forgotten the name. Iridogorgia?
Oh, over on the left. I didn't get a good look at it. Okay, that would
Find
figure.
And
this is a Walteria again, right in front of the rock.
What would you like to zoom in on while we're here?
The Walteria and the crab right on the top of it.
Zoom in, please.
It's not a crab. Steerike one.
It might be a crab. I see a
Yeah, it's
crab
a sea
in
star.
there.
It's a sea star.
Down lower, but there's a sea star. There's a brittle star, a basket star.
A basket star. There's a crinoid, I
on the
think,
back
on
too.
the
Yeah.
backside.
Yeah, there is. Right.
Okay, come wide, please.
And there's a little shrimp or a little crab in the middle of that.
This kind of illustrates how important these animals are for other animals because they're all glommed onto the sponge to get up off of the substrate. That's their perch, and they're all kind of sharing it very nicely right now. I think humans could learn a lot from these things.
You can leave bubble on the front porch, whatever normal setting. I don't need it. Oh, come on, just one. Oh, you. All right.
Why is it brown? Because it's rusty. Why do we put rusty plates down? Because they will eventually decompose.
It's hemp line, fully biodegradable.
It'll take a little while, but it will decompose.
Okay. Let's go look at that big thing.
Yeah, I agree. Okay. Oh, there it is. It's beyond it. Holy guacamole.
This is where the guacamole is, that's for sure.
And look at that other rock. A whole bunch of stuff on that other rock, too.
That's a really big sponge off to the left there.
Yes. Where is it? Oh,
yes!
We gotta go see that. Skip the Iridogorgia.
You can keep the ship moving. We're at the end of our leash, so we're gonna enjoy this Iridogorgia.
Okay, we will.
You know, cause
we finally see one of the living monster
the monster sponge.
I want to go down that way. Can we go down that way?
Let's go down that way.
All right, let's go that way.
Real. We actually saw a live one.
Bridge nav.
That's incredible.
Can you find it in your gallery, please?
Oh yeah, we were gonna look at this, right?
30 meters 150.
Steve said we might be good area for another Niskin for ED.
That's a very good point. Over there where those.
Roger.
I have to see what these corals are.
Stand by, Aaron.
It'd be kind of nice if we got a little cluster of a different.
No, sorry, stand by video. Double Aaron is tricky.
This only has branches at the top, but the spiral is not very tight. So I would think this is something like Iridogorgia superba, but
start with the base and work our way up.
Wow, that looks like that's a little Chrysogorgia.
I can go in further if you want to see it closer.
Okay.
So that's just the way it forms. It's not stripped by a sea star.
No. That's just normal. Yeah, I guess it loses its branches, right, or stops growing side branches at some point.
So pretty.
That is good size, but Yeah, with the not very tight spiral, I'm surprised you're not seeing more branches on this thing further down.
Mm-hmm.
The size of it. Okay.
Beautiful. All right, thank you.
How long do you think this is?
Well, those green lasers are 10 centimeters apart, so I'll paint them on the ground just below it. Stand by.
What's that thing floating up above?
Go. Thing floating.
There was like a red thing. What's that?
Oh, yeah, up there.
Yeah, I see it there. Yeah.
Spawnophore or something.
It's over a meter, maybe somewhere between 1 to 2 meters or something. I don't know.
You can try to zoom on that red thing. Oh, where am I
That's
going?
pretty cool.
Yep.
What are you?
It's
Yeah,
a salp.
So
Did it self?
Oh,
Yeah,
so
colonial
cool.
salp. So each of the red dots kind of marks where each of the actual animals are.
Wow. I want these lasers off.
The little LEDs. It's cool.
Well, that
Okay, come wide, please.
Steve's got a good point. This little rock here has got a different group. It's not quite as many of those Metallogorgia or the Remulogorgia. Got some more chrysos. I think there's a bamboo in there.
We're moving that way, so we can
Yeah.
check it out.
Yeah, I'm at the end of my leash.
Okay.
what's that bright yellow?
Look at that
underneath
sponge. Do
the,
sponge
that is
rules or
incredible,
what? They
yes.
rule.
They do.
It's amazing seeing it in the water. You know that would not be able to hold that weight in air.
Uh-huh.
We need
Yeah.
sponge rule shirts for this watch.
Mm-hmm.
That has got to be the biggest one of those guys I've ever seen.
Pretty clearly defines which way the current's going.
Looking at this rock feels like the previews before the movie.
Yeah.
Yeah, 135. Should I just
That
move
what?
them directly east so that it kind of holds off a bit from that?
The sponge?
Sure.
You mean the one in the background?
Sure.
Bridge
Oh.
nav.
Seems like the exciting part.
Can we change our bearing to 090, 15 meter step? Yep, thank you.
So you're talking about this guy, right? The coral? Yeah, Iridogorgia. It's not superb. I kind of screwed that up. It might be Magnus spiralis, but I don't know. But I think just leave it as Iridogorgia in your notes. Would be okay.
Steve's saying the bushy Chrysogorgia are intriguing.
Yeah. It's just a beautiful scene.
It is spectacular.
Yeah, Steve
Nice.
is noting this really bushy Chrysogorgia
Hey,
right
what's
in
up?
front here, I think. And
Hmm.
there's one down below that they're a bit different. They're pretty interesting.
Mm-hmm. Look at the size of that thing on the Argus
Yeah.
view.
Oh,
Oh, this is it.
Are you happy for me to
Agonizing.
zoom in, Trevor, on Argus?
Yeah.
Cool.
Yeah.
So I'm going to ask Steve a question here. Do you think those bushy Chrysogorgia are something that's worth collecting? I mean, I can't come up with an ID right off the bat. I don't know if you've seen them before. I've seen bushy Chrysogorgias, but might be something a little bit different. I don't know whether to help with Meredith's eDNA study or not.
Oh,
'cause-
actually, that's a good question there, Chris. If you want to do a Niskin, now would be a time because I can't move anyway. If
well,
that's of
yeah,
interest.
let's do a Niskin here.
Okay, I'll just touch down on this rock.
This is such a smorgasbord of different corals though that I don't think we really could collect one thing, and it wouldn't really help her out. We've got bamboos, coralliids, chrysogorgids, anthomastus. But yeah, let's get a water sample on this. She's going to get some DNA in there no matter what, somebody's DNA.
So I just touch down on this rock, that'll be
Well,
happy?
I think it's got to be a little bit above the surface of the rock, a little bit in the water column, I think.
Okay.
Yeah, while you're still sort of drifting.
Sure, yeah. I can just try to park the port side above it. I have
Okay.
to hover in autos, but that'll be fine.
But that sponge it almost looks like it's double the height of the ROV when you look at the Argus view.
That very well could be.
Yeah, and let's get the laser, when you can put the lasers
I mean,
on
the lasers are going to be
top.
unuseful.
Well, on the top, see how big the bell is.
That's true.
Okay.
It
You're
should
good to
have
go if you
DNA from different organisms that are around there.
Excuse me?
Say again?
The water that we're collecting.
Yeah, that's
should
the
have
hope.
Hydraulics
DNA
are
of
off,
different
means you haven't pressed the
organisms
blue
around
button.
there.
Yeah, because a lot of
Yeah.
these corals will shed
Yeah,
sort of
it'll
tissue and mucus and things that's going to have DNA in it.
I can help it, but just think of the amniotic fluid samples that some
Zoom out a
pregnant
little on Argus,
women get
please.
when they're trying to do a
Yeah, sure,
karyotype
all the way.
of
That's good.
their
Yeah, no big deal.
baby. It's like we're taking water from the womb of the ocean.
Yeah,
That's just my own thinking.
you know, this is a very early stage of this kind of technology development, what she's trying to do here. But it's a really interesting direction. I mean, and it's very intriguing how this is going to evolve as people get better and better at it, and we learn more about it. And after she gets more and more samples.
Yeah. I feel very fortunate to
Okay, hold on, sorry.
see this.
I forgot to step. Okay, so you're going to do anything but the top one, is my understanding. So 2, yellow one, do that.
Yeah, we got 2, missing 2.
2 is open?
Yep.
Okay.
Yeah, so these Niskin bottles basically are a way of collecting water from this particular area that we're in. And I think one of the reasons why we're interested in doing a sample here was because we saw such an interesting diversity of corals, and including this large sponge. And so what they're going to do is take some of these bottles back, take some of this water back, and analyze it for the DNA in the water column, which would basically allow
So when
us
you
to
get on, you can
categorize
actually
some of
twist
these
the jaws and
animals,
kind of wrap it up in
compare
there.
them against the database of
One of the
the
tricks.
eDNA that we have, and see if we can identify some of the organisms in it, contribute to that overall catalog. It's a really interesting process, a way to look at species in a given area without actually observing or collecting them. So do you know who does the DNA processing for that?
Well, it's Meredith herself in her lab, and maybe students or something. Or I don't think she has students, or maybe she does, but she's the one who's going to.
You got it.
I believe.
It's being elusive.
It's very tangly. The yellow one's a very tangly one.
Yeah, that blue one's interfering.
Yeah. The blue one's like, put me in, coach. There you go, yeah.
Got it.
Watch the camera. Hey, you got it. You can let it go. You got it, that's enough. Yeah.
Did it trigger?
Yep.
See, that's the trick. You think that's easy. Well, you got to get that 3D sense.
Yeah,
That's what's so hard, and that's very precise. You got to. So
So there is a comment here from one of our viewers about the 3D perspective. Has the team ever considered adding a stereoscopic camera with a VR headset to give the arm operator 3D depth perception?